Okay, thank you everyone, and I'm very happy to be speaking to you today. And I want to say that, we've all been celebrating Dr. Oberbauer this weekend, and so I do have to say that this talk about understanding breeds as populations evolved from a long email conversation that I had with Dr. Oberbauer over a period of time. So I want to thank again Dr. Oberbauer for making me put all of my thoughts into some kind of logical fashion to share with you today. There are lots of things I want to talk to you about today that I'm not going to talk about because my subject is understanding dog breeds as populations, but I still will throw some messages in here so that I do get some points across. I want to thank the Canine Health Foundation and Dr. Brown for inviting me to speak to you today. For full disclosure, I have no relevant financial interest in the products or services described in this presentation. I have taken no honorariums from food companies or other companies. I have not accepted research money from anyone, and so that stands. But I do have a lot of interest to disclose. So I am a member of the board of directors of the OFA. The OFA is the world's largest repository of genetic screening information on dogs, and also the largest DNA repository with associated health data. We don't have the most DNA on dogs, but we have the health data, and that is used for research. And so I'm very proud to be a member of the board of the OFA. I also am a breeder of Gordon Setters, and so in February, my bitch took breed at a little dog show, and then her litter mate Bobby, this was the day that he attained his junior hunter title, and we celebrated by spending 2 hours getting all the sticks and twigs out of him at that day. So these are my two litter mates that we're working with currently. Also, full disclosure, I am a solo small animal practitioner at Freshwater Veterinary Hospital. I'm not exactly sure what breed that silhouette is, but I do take care of dogs, and I do take care of cats in my practice. So I had to throw a little cat thing in there. And since we are talking about cats and dogs now, I do pollings. You need to pull out your phones and keep them on silent. But we're going to do some poll questions, and so on your phone, if you go to the web page, pollev.com/AKCCHF, this question will pop up. Or if you want to text, you text to the address 22333. You type in AKCCHF, it doesn't have to be capitalized, hit send. You only have to do that once on this poll, and then you're going to answer A or B. Hold on, we're not supposed to show that yet. And then you're going to answer A or B to your question. So either the website, pollev.com/AKCCHF, and just tap on dog or cat, or you're texting to 22333. One time only, you're going to send AKCCHF, and then you're going to do A send or B send. So we have 35 people that have answered, and you can keep on working on getting there, and those numbers are ratcheting up now. So I just want to make sure that we're in the right room here, just in case any of those other people have snuck in. And so you can keep on voting, but I'm going to reveal, and it shows that 93% of you are dog people and 7% are cat people. Where are the bloodhound people? We need some searching out to go on here. Okay, so, more people are answering, but I am going to switch to the next poll. Purebred dogs are all inbred. True or false? Okay, we're up to about a little over 80 people, so we'll show the results. And 2/3 of you feel that dog breeds are all inbred, and 1/3 do not. So that is something we are going to address today. And my last poll question to start things out, my breed has detrimentally limited genetic diversity. True or false? we've got over 90 people here. And so we'll show the results. So 58% feel that your breed has detrimentally limited genetic diversity, and 42% feel that that is a false statement. So that is something we're going to address today as well. Okay. By the way, I know that you received copies of the preceding article on this talk a few weeks ago, and mainly that was because our discussion session was yesterday prior to the talk, and I kind of said to Dr. Brown, well, you know, if I'm not presenting this information, how can we really discuss it? So that's why you received it. It is also in your proceedings, so don't try to write everything down that I say, because I'm going to start talking faster and faster, which is what I usually do. And it's all in there, and plus all of the talks in the conference are being recorded, are going to be distributed to you, so you can bring them back to your breed clubs as well. So I want you to sit back and pick up the concepts of what we're talking about. Dog breeds are like different ethnic populations of people. Let me get my little clicker out here. All people on Earth are humans, Homo sapiens, but we are not all closely related. Ethnic populations originally arose due to geographic isolation. So man originated in the Middle East, and due to population expansion went to Asia, went into Oceanic, to Africa, to Europe. Those from Asia went across into the Americas and down in the Americas. And so over tens of thousands of years, this is the evolution of man. There are some ancient mutated genes and hereditary diseases that are shared by different ethnic populations of people. So ancient mutated genes cause common disease that are shared across all ethnic populations. In some ethnic populations, certain common genetic diseases occur at a higher frequency. So some populations have more high blood pressure or diabetes, even though all populations have those diseases. Some ethnic populations are prone to certain genetic diseases that are seen rarely in any other populations. So they're only seen in a specific population. Dog breed development followed human migration and evolutionary paths. So if you look at all of the original work of Dr. Heidi Parker and Dr. Ostrander's lab, you know, we have a clade of the ancient Asian breeds, and we have a lot of different European breeds, and then if we look at the differences of some of the African breeds and some of the oceanic breeds, and then the few American breeds that were brought over by the migratory human populations, we see the migration and then the development of these different populations. So Canis familiaris, the dog, was developed to live alongside man, honing their natural skills of hunting, guarding, hearing, so scent barking, scenting, and protection. Common genetic disorders are caused by ancient disease liability genes that preceded breed formation. So these are shared by many breeds and by mixed breed dogs. And so this is from a figure in a publication by Dr. Shaston Lindblad-Toe at the Broad Institute that showed multiple founding events of the domestic dog out of the wolf populations, and then how they evolved down into groups of dogs and then into individual breeds. And so genes for allergies, the most common genetic disease of dogs that we see every day in veterinary practice, those genes are ancient genes that have trickled down to just about every breed. And hip dysplasia is very common. And then only some breeds have patelloluxation. Some due to morphological conformation, being the small breeds, but there are other large breeds that have patelloluxation as well due to common inheritance. And aortic stenosis, as Dr. Stern so eloquently was talking to us about, we see in certain different breeds that have some commonalities. So common canine genetic disorders, allergies, hip and elbow dysplasia, inherited cancers, patelloluxation, non-struvite bladder stones, because struvite stones are environmental from infection. All the others are metabolic and inherited. Hypothyroidism, mitral valve disease, inflammatory bowel disease, diabetes, retained testicles, umbilical hernias. all genetic disease, all common genetic disease shared across most breeds. I want to talk a little bit about selection because all of the diversity, we talk about the plasticity of the dog and how much diversity there is in the species itself, but we need to understand how we created that diversity because the dog is a man-made creature. All right, so let's talk about selection. If this circle represents the entire gene pool of your breed and you want to select a breeding dog, but the first thing you want are certain characteristics that you want in your dog. So you're looking at the part of the population that has those characteristics, and this part of the population that doesn't have those characteristics, you're not considering in who you're going to select for breeding or select for a stud dog to breed to your bitch. And then you add something else. There's a genetic test for a very serious disorder that we have, and we don't want that disorder, so we apply that on our selection index. And then there's something else that we either want or that we don't want that is added to that selection index. And then there's another genetic test in genetic disease. Even if there's no test, there's screening for it, for hip dysplasia or even behavioral pathology or other types of things. So we add that to the mix. And what happens is there's only a very small part of that population that you're actually considering dogs to use if you're using all of those selection indices in your decision making. And I could go on for hours, and you guys have heard me in your breed health seminars at your national specialties go on for hours about a lot of these things in breeding theory, but the bottom line is that every single time that we select, each thing that we add for selection limits our choices. And therefore, if we're using things for selection, they darn well better actually represent what we think we're selecting for. So the inappropriate use of selection, the inappropriate use of genetic tests, or inappropriate genetic tests is a very important topic if it limits our ability to actually select for what we want to do. So breed formation and chromosomal inheritance. Let's talk about those because we hear a lot of words about genetic diversity and homozygosity and a lot of terms that we need to define for you. The development of breeds occurred through artificial selection for body type, color, coat type, behavioral, and conformational aspects. As breed lines became more specialized and stud books closed, those who did not conform to the standard or who exhibited health issues were removed from breeding. And that's how we developed our breeds. So many of you who have seen me break down your breeds gene pool and pedigrees and analyze that for you to talk about your breeds are familiar with this type of figure here. This is a pedigree of a single dog. This dog right here. And his parent, this is a pedigree that you're used to seeing at this end, okay, the parents and grandparents and so forth. But these are the founders of your breed. And many breeds were founded by a very few individuals, except for those of your breeds that had a working phenotype that started your breed, which tended to start from a larger group of individuals that were able to work in a certain situation, and that was the phenotype. But then we still went through a bottleneck when we wanted them all to look alike and reproduce the same, and so that you then had a small population of founders. And throughout this area of the development of your breed, there was selection, there was purging of those that did not conform or that were not healthy, and this was how your breed formed. And once we had a breed that reproduced to standard and that was healthy and was what we wanted, we then go through this population expansion phase where that population grows. And so these are all the individuals behind this single dog. If you're looking at the entire population, it actually continues to go all the way up there and all the way down there to include all of the individuals in your breed. But because we're looking at the pedigree of a single dog, we then truncate once again to that individual. So as only a small number of dogs are used to produce the next generation, rapid change can occur in the breed's genetic background. In many breeds, in your larger population breeds, less than 5%, and in some breeds less than 2% of the generation is used to produce the next generation. In some of your smaller population breeds, that percentage is much higher, and it actually makes it even more important that you do proper selection with a small population breed, because each litter has a larger impact on your breed. Those individuals who demonstrate and propagate desirable characteristics will have an increasing influence on the gene pool through multiple generations of descendants. If an individual has quality, produces quality, and their offspring produce quality, they will continue to influence your breed, and that individual and his or her genes will have increasing influence in your breed. Dogs that produced offspring that were unhealthy or inferior to a standard are not used, and their influence and that of their ancestors is diminished. So that's how ancestors move out of being influential from a breed. So all breeds have several, when you analyze a complete pedigree database of a breed, which is what I do for every one of you that when I do these presentations, all the way back to founders, we find several influential ancestors that appear far back in the pedigrees, but pass on a high percentage of their genes to every single individual in the breed at very high frequency. So for instance, here's Cavalier King Charles Spaniels. Daywell Roger was born in 1945. But he contributes almost a third of the genes to every single modern Cavalier King Charles Spaniel. Okay, he is the most influential dog in the breed. Cannon Hill Ritchie, Anne's son was born in 1927 and contributes more than any single grandparent to a Cavalier King Charles Spaniel. Here we have bearded collies. And so what we're looking at here is through the decades, the influence of individual dogs. And so as you see, Bailey of Bothcanar, his influence continues to go up every single decade. So the quality that Bailey of Bothcanar's descendants carry continues to increase his influence on the breed. Whereas other individuals, their contributions go down with each generation because their descendants are not passing on the quality that you're looking for in your breed. And these influential ancestors are not necessarily founders. The founders here are marked with asterisks. Bailey of Bothcanar was just a dog that came along in the 1940s and that was of quality, produced quality, and therefore his influence continued to grow. When we look at a pedigree, and this is a pedigree of a this is a pedigree of a Cardigan Welsh Corgi that is relatively outbred. And when we look at a 10-generation inbreeding coefficient, so only looking at the ancestors back to 10 generations, this girl, Sybil, has an inbreeding coefficient of 6.3%. And that most of us would consider outbred. In a 3-generation pedigree and even a 4-generation pedigree on this bitch, there are no common ancestors between the sire and the dam. But if you look all the way back to founders and calculate the inbreeding coefficient, which represents the amount of homozygosity in the dog, it's 23.1%. And as we know, a full brother, full sister mating or a father-daughter mating has an inbreeding coefficient of 25%, and we consider that inbreeding, highly inbred, where in actuality, every member of this breed has that kind of inbreeding coefficient. And every member of every breed is going to have that kind of inbreeding coefficient if you have analysis that goes all the way back to founders of the breed. So when you look at a pedigree analysis of Sybil, the most influential individual is Withy Brook Brock, who doesn't even appear until the 14th generation, appears over 500,000 times, and contributes as much as any grandparent. And you look at Bob Lloyd or Cassie, who each don't appear till the 17th generation, but each appear over 28 and 29 million times in the pedigree, because that's how many pedigree spots are going to be present in that many generations, because we're going back 30 generations or 50 generations when going back to founders. That is how the background of your breeds developed, and that is what every single breed is going to look like this, especially looking at an outbred individual. You look at a line-bred individual, they're going to have names up here with high percentages that only appear for the first time in the second or third or fourth generation and only appear a couple of times. So actually, the line breeding on this dog here that I can't pronounce because I'm not Welsh, did not appear to the fifth generation, appears 11 times. And that would be considered a line individual who was line bred on. So here's one of my funny cartoons that I like, Get Fuzzy, and Rob says, ever heard the expression familiarity breeds contempt? And Satchel sits there and ponders and he says, no, in the dog world we say familiarity breeds hip dysplasia. So this is what the public thinks about purebred dogs, is that they're all inbred, and because they're inbred, they all have disease. So dogs have 39 pairs of chromosomes, one in each pair from its sire and one from its dam. So here are the chromosomes paired up, and one from the sire and one from the dam, and you've got your X and your Y chromosome there. So chromosomal crossovers during meiosis, so we learned it in high school, so you gotta go way back, is that meiosis is when you're forming sperm or eggs. And in meiosis, as those form, there's a crossover that occurs between the chromosome from your sire and your chromosome from your dam, from your mother and your father, and they can exchange material. And that's what causes entire chromosomes to not be inherited from one generation to the next. It mixes up the genes on the chromosomes. And so here we have, an egg and a sperm from the parents going to this dog. And so then when this dog reproduces and goes through meiosis, those genes are going to, those chromosomes, and we're just looking at one pair of chromosomes, and they're going to exchange genetic material through crossovers, and then you now have a mixture of chromosomes from the sire and from the dam into the sperm of this dog. And so that causes the mixing of the genes. So the big piece of chromosome that came from one of those parents is called a haplotype block. And a haplotype block is just a whole group of genes that got inherited together on a piece of a chromosome, okay? And so if you're selecting for a gene in that block, you're also selecting for all the other genes in that block, and that's called linkage. Those genes are linked to that selected gene that you're selecting for or that you're selecting against. But in actuality, because we're looking at a big block from the sire or the dam, well, that block actually includes a lot of different segments of chromosomes from different ancestors from the background as well. So even though it's a block, it does contain genes from many different dogs. Selection for positive traits will cause the inheritance of a chromosomal segment from the parents containing causative genes. And that sperm is very happy because it's going to contribute to the gene pool in the next generation. Selecting against deleterious traits or diseases will cause the loss of a chromosomal segment containing causative genes. So this is the action of selection. As crossovers occur through the generations, the size of the chromosomal segment containing genes under positive or negative selection can get smaller. So as you have more crossovers occurring over generations, that haplotype block or segment of chromosome that these molecular geneticists are working with can get smaller. When you line up all the dogs, you're going to see, for a selected gene, that area that is going to be the same because of selection and what we are looking for. So there is your haplotype block there. As dogs are line bred on, appearing in both the sire and dam side of the pedigree, this can cause haplotype blocks to pair up, causing what molecular geneticists call runs of homozygosity. So homozygosity is the pairing of like genes on the chromosome from the sire and the chromosome from the dam. And so when those genes have haplotype blocks that are included, and they pair up, we have runs in that haplotype block of homozygosity containing the genes in that block. So that's what that term stands for. Breed-defining genes would be expected to be collected in runs of homozygosity due to selection over time. These include breed-selected traits for size, coat color, texture, behavior, skeletal morphology, and other traits, whatever you are selecting for. Even without close line breeding, selecting for positive traits according to your standard and for health will increase the homozygosity of these haplotype blocks having originated from distant ancestors. So it isn't even active line breeding that causes this, it's just selection within our breeds. So I want to just make a couple of statements here, all right? So dog breeds are inbred, okay? We have selected. They are clones, all right? When you walk into a dog show, you know exactly what breed is in this ring and exactly what breed is in that ring because they all look alike, all right? And so that is what we have done with our breeds purposefully. We have done it on purpose, okay? It wasn't a mistake that we've created homozygosity and runs of homozygosity in our dogs because we want them to look alike in that ring. And we love our breeds for their individuality. So this is what we see. So let's talk about the downside, diseases and disorders. And watch for it, the dark side of breed development. How do breed-related deleterious genes accumulate? They can accumulate many different ways. One is for direct selection for disease-associated phenotypes. If we select for something that predisposes for disease, it's no surprise that we get that disease, okay? We can have linkage to selected traits, so genes that are in a haplotype block to a selected gene that are just moving along with that gene. We can have deleterious genes that are carried by popular sires that increase in frequency because they are used frequently. We can have genetic drift, which is just the random movement of genes in the background of our breeds. Or, very importantly, we can have the absence of selection against deleterious phenotypes. If we don't select for health, we're not going to get health. And that's an important message. Let's talk just a second about purposeful selection for disease liability because it's a hot topic around the world. We have breeds being banned. Pugs are banned from being bred in Denmark now, and we're working with the legislatures so that they can understand the science behind this and that's not the right way to do things. You know, and when we talk about the flat-faced breeds, the brachycephalic breeds, these are some of the most beloved breeds in the world. They are not going to go away because some legislative body decides that they should go away. We love them, they will be bred, and they're wonderful dogs. And you know what? The majority of those breeds are healthy, but we have a lot of dogs that are not healthy, and so what we need to do is to scientifically select away from some of the extremes. And it's not just brachycephaly, it's excess size, it's excess skin, it's excess angulation. There are so many things that are good in a healthy dog and get bad when we stretch it to an extreme. So most breed standards do not call for the expression of extreme phenotypes. And so what we have to look for if we are seeing a breed-related disease due to extreme selection is to move to moderation away from those extremes so we maintain that phenotype, but it becomes a more healthy phenotype. And it's important that we do that through selection and breeding. And it's very important that we have judges' education and we educate the judges not to select and award the extremes. Because that is part of the engine of the fancy that whatever is rewarded is what we're going to breed. And so we all need to recognize that our dogs are beloved and they can all be healthy, but we need to maintain their health. All individuals carry some deleterious mutations. Quality individuals who propagate will also propagate their deleterious mutations, and those can cause breed-related disease if they are disseminated and increase in frequency through the founder's effect. And we see this not just in small population breeds, but in the largest population breeds, that founder's effect and popular sires can overrun a breed and introduce new diseases that we've never seen before. Individual liabilities can cause embryonic death, resulting in smaller litter size or infertility, increased neonatal death, or breed-specific genetic disease. Individual liability genes cause that. It's disease-causing genes that cause these things. It is not a function of homozygosity or of inbreeding, okay, but of the accumulation and propagation of specific disease liability genes. So the molecular genetic studies of breeds that the molecular geneticists do, they line up all the chromosomes and they show the haplotype blocks that they find, and it exactly mirrors the pedigree-based inbreeding coefficients and influential ancestor coefficients that I've been talking about to you. So it is not surprising to us as breeders and as people who study pedigrees and understand the backgrounds of our breeds that they're finding large haplotype blocks and large runs of homozygosity and increased homozygosity within our breeds that are making them say, oh my God, we have to help these breeds and make them healthier by reversing what has been done for generations, what we purposefully have selected for. These are necessary and expected consequences of breed formation and evolution. Homozygosity is not inherently correlated to impaired genetic health, nor does it need to be artificially controlled. So to understand that, we need to understand about natural species, because in actuality, all of the recommendations that are coming to us about homozygosity were developed based on studies of endangered species and endangered species population geneticists. Any genetic change that improves the chance of survival and the ability to reproduce will increase in frequency in species evolution. So that is the biological imperative. Thrive and reproduce. That's the only thing you have to do to survive as a species. Lines that are disadvantaged will diminish in influence and die out. Any selection, though, whether natural or artificial, will cause a loss of genetic diversity. And this loss is not detrimental to the population if it's directly related to increasing its superiority. So we do lose diversity through breed development, but that is not detrimental if it causes superiority. Genetic disease in species occurs primarily through genetic drift, the random accumulation of disease liability genes in the absence of selection. So that's what we deal with in natural species. Healthy carriers of recessive and additive disease liability genes propagate these genes in the population. So again, the differences between natural selection and artificial selection. Natural selection, all individuals have an equal chance at reproduction. And I understand that species behavioral structure, there are dominant males with harems, but there are also those scoundrels on the periphery of the population that, that also get to impregnate females and contribute to the gene pool. And there is no selection. It's only natural selection. Artificial selection, only a small percentage of the population reproduces, which is why it's so important who we select for reproduction, and there is selection for specific traits. So the species survival plans designed by these conservation population geneticists were developed for species that have a limited number of breedable individuals. They mate the most unrelated individuals together to avoid homozygosity of deleterious recessive genes. And that is all that those plans are about. They also want to maintain the breadth of the genetic diversity of the population by finding individuals that are the least related to others and maintaining them in the population. Even if they're unhealthy, let's use their unique background in maintaining the diversity of the population. And that's important for both artificial and natural selection, maintaining the breadth of the diversity of the gene pool. When we analyze entire breed population databases back to founders, Every dog breed, regardless of its population size, has the same population genetic findings. High, deep pedigree, average inbreeding coefficients or homozygosity, low effective population size, a limited number of founders or number of ancestors contributing to diversity, and high average relationship coefficients to individual influential ancestors. Breeds with small populations look the same as breeds with large populations did much earlier in their evolution and development. So the difference between saying, well, Labradors are much better because they're much more diverse than a small population breed, well, you know what? And this small population breed is on the verge of going extinct, and Labs are going to be just fine. You know what? Labradors looked like that a long time ago, and look what happened to them. All right? Selection of breeding animals should represent the quality traits and breadth of pedigree background. Quality lines should not be abandoned. So the popular sire syndrome is one of the biggest aspects of limiting genetic diversity. And so here's a geneticist's pedigree with circles and squares and matings, and a male comes along and everyone loves him and they ooh and ah, and different bitches from different areas of the gene pool are brought to him, and everyone likes what he's producing. It's looking great, and so they start line breeding on him, and very quickly his genes are distributed across the entire gene pool. But the overuse of a popular sire quickly disseminates his genes throughout the gene pool, and this is the important point, without the benefit of evaluation over time. And the more insidious aspect of the popular sire syndrome is that it causes the loss of other quality male lines that should be used. And they're being sidelined because there are only so many quality bitches that are being bred in each generation. And if an overabundance of them are going to a popular sire or a popular sire line where you have a sire and his son takes over and his son takes over and the entire breed truncates on a single popular sire line, that contributes to our limited gene pool. A popular sire's influence is different from that of an influential ancestor. An influential ancestor's contribution is continually evaluated through its descendants to compete with others for breeding status. So every generation, influential ancestors' descendants are evaluated to say, this is a quality dog, we're going to use it for breeding. Whereas a popular sire, we don't have that evaluation over the generations. Everyone's just breeding to him. And another insidious aspect is once we realize that a popular sire is carrying a lot of detrimental things that we don't like, when we try to eliminate the influence of that sire, we are now eliminating the influence of an entire generation of quality bitches that were brought to him at the same time. So a huge influence on our gene pools. So healthy gene pools require expanding or large stable populations. So when you have small population breeds, what's the most important thing? Keep breeding. You know, if you're afraid about genetic diseases, we try to deal with them, but you make the best possible choices and you keep breeding and expand your population. It expands the choices that you have in the next generation. It allows creation of new family lines and within breed diversity. So lastly, I want to talk about genetic diversity because it is a hot topic because it has spawned the development of diversity breeding. And so we need to understand this, and a lot of people have questions about this. So in response to perceived concerns with genetic diversity, some advocate for species survival plan type of outbreeding programs, recommendations to only breed to those least related. And what that does is it homogenizes breeds and erases the genetic difference between individuals. If you're looking to find the most unrelated individual to breed to, you breed to them, the offspring are now related to each other. So now you need to find someone else that's unrelated and breed to them. Now they're all related to each other. So now you have to find someone else that's unrelated and breed to them. Now they're all related to each other. You're going to run out of unrelated individuals, and everyone's going to be homogenized in the center of the gene pool. And you don't have individuals at this part of the gene pool and that part of the gene pool anymore. And what is selection? Selection means identifying differences between individuals. So I don't want this, I want that. But if everyone's right here, it eliminates our ability to select. And we do have, and I'm not singling out any one company, but there are several companies that are promoting these genetic diversity indices and telling breeds and breed clubs, you need to use these to increase your genetic diversity and to make your breeds healthier. But breed diversity requires distinct lines, and I've already talked about this. Outbreeding will not change the frequency of deleterious genes. For dispersed breed, if you have breed-related disease, those genes have already been dispersed. So whether you line breed or whether you outbreed, the chance of pairing up recessive genes is going to be the same, and you will have the same frequency of affected individuals in your breed. The only difference is that they will appear in a random fashion rather than a predictable fashion. And if you want to know what that looks like, look at our cat populations, because 95% of America's cats are random bred, but we see inflammatory cystitis and diabetes mellitus and gum disease and inflammatory bowel disease all day long in these outbred cats. Randomization of genes in the population is the exact opposite of selection. It does not support breed quality or health. Recommendations for selection to increase heterozygosity or minor allele frequencies or minor frequency chromosomal segments do not select against disease liability genes. And I understand the molecular geneticists are trying to help you out, but they have no idea what genes are represented in those chromosomal segments or in those minor frequency alleles. And in all likelihood, those became minor frequency from generations of selection against them and selecting for those major frequency alleles or haplotypes to create those runs of homozygosity that allow your breed to be healthy and breed true. So following those may reverse the efforts of positive selection. Recommendations to increase the diversity of MHC haplotypes. I talked about this in the roundtable last night. No breed in no breeds has general limited MHC diversity or random homozygosity been shown to impair breed immunity. There are not breeds that are constantly coming down with infection due to just homozygosity. And all studies of autoimmune disease, immune deficiency, pathogen susceptibility identify specific MHC haplotypes or disease-related genes. So again, it's detrimental genes or detrimental combinations of MHC genes that cause disease, not their homozygosity, not general homozygosity or limited number of MHC alleles within a breed. When breeds show fixation of deleterious traits or significantly diminished fertility or fecundity due to genetics and not environmental causes, which we won't talk about today, they may require an SSP-type plan or to open the stud book, to importation from other populations, because you know what? The molecular geneticists, they can tell us if a golden retriever is from America or the UK, because they are different populations, and they can be separated on their molecular basis. They can tell you if a dog is a conformation dog or a working dog or a field trial dog, because they are different populations, but they are the same breed, and they are a source of genetic diversity in your breeds. Or possibly crossbreeding if a breed just doesn't have the diversity for healthy genes for specific diseases to bring in those new genes. But most breeds do not find themselves in this position and only require proper selection and improve their gene pools and genetic health. So managing breeds requires breed conservation, not species survival plans. And if you want to understand about breed conservation, I suggest that you get Dr. Spanenberg's book, Managing Breeds for a Secure Future. He has worked with legacy farm breeds that are limited in their numbers and how to expand their populations and improve them. And he has included dogs in the latest edition of the book, and there's a lot of helpful information in there. Breed health should not be judged on statistics of inbreeding coefficients or homozygosity, but on breed health surveys. That's why it's so important. That's the monitoring for your breeds to understand, are we healthy, are we not healthy? It's not based on molecular genetics. It's based on, are the dogs healthy or not? Okay? No one's going to tell you that your dog is unhealthy if it's living until it's 15 years of age and it's never on any drugs in its life and it's performing and it's doing a great job. I don't care what the molecular geneticists are going to tell you about that dog. It's a healthy dog, okay? A healthy and diverse breed gene pool should have outbred clusters as well as different line-bred families, so it allows us selection. And that's how you create a master hunter like my dog Smitty here. Genetic diversity is breeder diversity. That is my mantra. If it's the varied opinion of breeders as to what constitutes the ideal animal and their selection of breeding stock that maintains breed diversity. So I'll be honest with you. If some of you want to do diversity breeding, that's fine. And some of you want to line breed on certain lines, that's fine. And some want you to line breed on other lines, and that's fine. And some of you want to bring in something you don't have and you outbreed, and that's fine. And then the next generation you want to go back and solidify it, and that's fine. But to say to everybody, you must do diversity breeding is wrong. To say to everyone, you must line breed on this one line because it's the superior line in our breed is wrong. Everyone needs to do something a little different. So lastly, I'm going to go quick on improving breed population health through health-conscious breeding, because that's the most important message, that we need to select for health. Early on, breeders judiciously removed animals from breeding that were unhealthy, could not excel, or did not conform to a healthy standard. Somewhere along the way, the responsibility to select for health and produce healthy offspring disappeared from breeding. And I don't know why that happened. For those of you that are going to have kids or are having grandkids, if I look at the age of some of us out here, what is the number one thing we hope for our children and our grandchildren? That they're healthy, okay? Without any question. Maybe we want a boy or a girl, but that they're healthy. And that has disappeared from dog breeding. People just breed and expect healthy offspring. Selection needs to be intense. If you relax selection, you will not get what you want and you will not get health. If artificial selection does not select for health or selects for deleterious phenotypes, then there could be no expectation of genetic health. The genetic health of the breed will decline. Genetic screening is a requirement and not a choice. I'm just going to finish up with a couple of slides here. Without evidence of pre-breeding genetic screening, health guarantees, because that's the big thing. Well, I don't do genetic screening, but we have a health guarantee. Guarantees that provide for a replacement of a family member once the emotional bonds have been made are worthless. A dog is not a toaster, okay? It's only a piece of paper written to excuse a breeder from performing their ethical responsibility of pre-breeding health screening. So who's a reputable breeder? It's one that does genetic screening. If not, you need to find a different hobby or profession. You need to do something that you can actually be good at. The proper use of genetic screening and genetic testing increases our breeding choices, okay? Genetic tests should not cause us to lose diversity because we're zealously selecting against carriers or zealously following instructions. It should increase our ability to use dogs that we want to breed that might carry deleterious genes. Official screening tests should be made available on something like an open database or a semi-open like the OFA, and so that's available so people can see you're a health-conscious breeder. In today's environment, not testing for documented breed-related disease is irresponsible breeding. The only way to select, going back to my original slide, against specific genetic disorders is to select against the causative or liability genes through direct genetic testing if there are valid tests, or phenotypic genetic screening if we don't have genetic tests. The most complex genetic diseases are complexly inherited, involving several genes and environmental factors. Most don't have a DNA test. Many have no screening or diagnostic tests, and they require medical history for diagnosis, bloat, allergies, epilepsy, and so forth. And breeding control requires knowledge of familial history of affected individuals. Pre-breeding health screening evaluates the health worthiness of the dog for breeding. If the public demands healthier dogs, this is where it must occur. So OFA has chick, AKC has bred by heart. Most of them are very similar. These should be realistic tests. Every breed needs to reevaluate their chick and their bred by heart requirements. Are they realistic? Do they include the important genetic tests that should be done, or do they have the tests on there that should be recommended but not required? Because that informs the public, it informs the breeder, and it informs the veterinarian of what issues could be occurring in your breed. So I'm just going to go to this last slide here. Pre-breeding health screening should become as universal as equine pre-purchase examinations. We need to change the culture of breeders and the public. The public should know to ask for evidence of health screening, of a health exam result on their breeding stock that says this is a healthy dog. It has been checked for heart disease. The history has been checked for other skin and musculoskeletal and other diseases, and it has had appropriate breed-specific pre-breeding genetic testing and genetic screening that has been done. And if we can change that paradigm, we will have healthy dogs. Thank you very much.